All right, we're ready to get started now, so if you could please take your seats. So welcome everyone to the first awards plenary session of the conference. My name is Kim Wickland and I'm the chair of the ASLO Awards Committee. Earlier this year, the 2017 ASLO Award winners were announced, and over the course of this week, we'll be hearing presentations from the award recipients. And before I introduce today's speaker, I first want to urge all of you to take a few minutes when you have a chance and go to the ASLO Awards website and read the descriptions of all the awards that we have. We have a really diverse portfolio of awards to honor achievements at all career stages and for specific activities within the aquatic sciences. And also, I'd like for you all to think of what colleagues you might have that you think are particularly deserving of an award, and take the time to nominate them, because really the nominations are the foundation of the awards program. They're submitted online through the website, and the deadline for the 2018 awards is this fall. So that gives you plenty of time to think about who you'd like to nominate for next year's awards. Okay, so today I'm honored to introduce to you the 2017 Raymond L. Lindeman Award winner, Dr. Shawn Devlin, who is an assistant research professor at the University of Montana Flathead Lake Biological Research Station. This award honors a young scientist for an outstanding peer-reviewed paper in the aquatic sciences, and is given in honor of Dr. Raymond L. Lindeman, whose seminal paper on trophic dynamics was published just after his passing at the age of 27. Shawn is receiving the Lindeman Award, oh, you got it, sorry, for his paper entitled Top Consumer Abundance Influences Lake Methane Efflux, which was published in 2015 in the journal Nature Communications. This publication is from Shawn's postdoctoral research at the University of Jyväskylä in Finland, where he worked with Roger Jones. Their study was a novel whole lake manipulation to test how trophic dynamics regulate food web and biogeochemical processes, and they showed that the emission of methane from lakes can be manipulated by altering the food web. And while the concept of such trophic cascades is not new, Shawn's research is novel in his combination in the study of the study of food webs, microbial ecology, and biogeochemistry to demonstrate the connection between trophic structure and inland water greenhouse gas emissions. And this award is particularly appropriate in that Shawn's work builds directly upon Raymond Lindeman's foundational research on trophic dynamics and works to extend Lindeman's view into the atmosphere. So please join me in congratulating Shawn Devlin.
Well, thank you very much for coming to listen to my presentation today, and I am honored to accept the award. I think we'll get started. If you could put my talk up here, and we'll get going. All right, so first I'd like to get out of the way some acknowledgments. I owe a great deal of credit and a great deal of thanks to my co-authors, Jari Suvaranta, Jata Sarenheimo, and most especially Roger Jones. So I feel like this work is really resting upon a 30-year career in methane and methane dynamics and food webs that Roger has really put forth, and I want to wish him the best in his upcoming retirement. So I'll really appreciate the opportunity to go to Finland and work with him, and I thank him for that. I'd like to thank ASLO and the awards committee, and it's a truly overwhelming honor to receive this award. I should note that and thank my institutes and various organizations in Finland, the University of Jyväskylä, and the Academy for the funding. I want to extend an appreciation and a thank you to Bonnie Ellis in particular for encouraging me when I was writing the paper up, and to Jack Stanford for the nomination. And I'd also like to thank all my colleagues and Jim Elser at the biostation, at Flathead Lake Biostation. It's truly a privilege to work there. All right, so we're going to talk a little bit about microbes in lakes and how they play a very important role. So starting out, they're a critical component to biogeochemical processing in lakes. They provide two big services. They connect the living organisms in the lake to the detrital organic matter in the lake via nutrient processing and as a active food web member. So by recycling inorganic or organic nutrients bound to organic matter into inorganic nutrients, they perform a vital ecosystem function and provide a critical role in terms of recycling nutrients and allowing them for phytoplankton uptake and autotrophic production. A little lesser known than the microbial loop that I just described is that they play an active role in the food web. So more and more we're learning that microbes are a key item of diets of zooplankton. And we're going to talk a lot about zooplankton eating microbes here in the next few minutes. Whoops. I'd feel pretty remiss if I didn't throw up Lindeman's plot here, because I feel like every time I think about this work in Finland and this paper, it really is, it reminds me of this figure, in that Lindeman put the bacteria right where they belong, in the center of everything. And they're really, I think that this work really ties into a lot of the concepts in his amazing paper. And it's truly an honor to receive this award. One of the metabolic processes that bacteria provide or use in lakes in particular is methanogenesis. So methanogenesis is something that is at the end of the road in terms of organic matter degradation, and it really is a key process in turning organic matter into bacterial biomass and into methane. So methane as a byproduct of organic matter degradation is a pretty major process, and it's found in a lot of systems within the guts of ruminant organisms like cows, in organic soils that happen to be waterlogged and become anaerobic, and in rice production is another big source of methane. But there's a few processes that happen in terms of how methane is formed, and it's system dependent. So the right biogeochemical factors have to be there, and it's mostly an anaerobic process. We're learning a little bit more about aerobic production, but still lots to be learned there. Because methane is a really powerful energy source, there's a whole other group of organisms that have learned to use it as an energy source and to hone in on that resource. And these are methanotrophic bacteria. A very common place where biogenic methanogenesis occurs is within hydrovents in the deep ocean, and a little bit more commonly found in lakes and fresh waters. So wetlands, lakes, and rivers have all been shown to be significant sources of methane and have a lot of active methanogenesis taking place. Another the group that has come to rely on methane as an energy source is methanotrophic bacteria, and these methane oxidizing bacteria, or methanotrophs, utilize methane as an energy source, and they really drive whole ecosystems in some cases. Tube worms, for instance, can be 100% reliant on methane as an energy source, and a completely chemotrophic energy pathway developed. And although we don't have flashy, nice tube birds in fresh water, we have the freshwater equivalent in chironomids. So chironomids have been known to actively garden methanotrophic or methane oxidizing bacteria by creating the perfect biogeochemical habitat for methane oxidizing bacteria to grow, and then use them as a resource. So in lakes, in the profundularian lakes, we see that some methane or some chironomids are entirely supported by methanotrophic processes and through chemotrophy. So in this case, we see that the chironomid pulls in oxygenated water, creates a very nice oxic-anoxic interface within the tunnel or in the tube, and they grow methane and harvest it and eat it and rely very heavily on that carbon. The conditions that sort of dictate whether or not methane is an important energy source in lakes is very dependent on the physical nature of the lake. The development of stratification, the anoxic oxic boundary layer is a really important role and regulates how much methane can actually incorporate into the food web. So in systems where there's no hypolimnetic anoxia, in lakes anyway, with no hypolimnetic anoxia, there's really not much methane that makes its way into the food web because there's not a lot of exchange between methane and organisms. In lakes that have intermittent hypolimnetic anoxia, you can see that sometimes profundal, like I described with the chironomid, that we do see methane finding its way into the food web based on this. But really when you have a perfect condition, which is a prolonged anoxia in the hypolimnion, you end up with prime conditions for methane to make its way both into the benthic or profundal food webs, but also into the pelagic food web. So when there's extended and long-term hypoxia or anoxia in the hypolimnion, there's an oxygen anoxia at the anoxic boundary layer. Methane oxidizing bacteria can really thrive and provide a very important resource to zooplankton. It's not just a spatial artifact of how methane can work in, it's also a temporal, and it depends on the concentrations that are found in the lake. As the methane builds up in the hypolimnion and slowly diffuses into the epilimnion where zooplankton can use this resource, the reliance of methane increases. So as the methane builds up, becomes more available, we see increased utilization by zooplankton over time. So there's a temporal dynamic too, it's not just spatial. The fate of methane is sort of dictated by the factors I just described, but as well as where that methane is a product of the sort of balance between methane oxidation by methanotrophic bacteria or anaerobic processes and methanogenesis. So whether or not a system has methane that effluxes to the atmosphere is dependent upon the balance of those two processes. Sometimes methane can be released to the atmosphere mediated through plants, and this is the case in rice agriculture. I think hello? Can you guys hear me? Okay, good. Sounded like the mic went up, sorry. There's also a lot of methane that leaves the lake through bubbling or ebullition. The processes that incorporate into the food web, like I said, depend on whether they're weakly stratified or heavily stratified, but ultimately the factors that dictate whether or not methane is leaving the system as a atmospheric gas is dependent upon the balance between methanotrophy and methanogenesis. That balance is pretty important because on a global scale, methane or lakes provide a lot of methane to the atmosphere. So inherently, the balance of methanotrophy versus methanogenesis dictates how much methane is leaving freshwater systems. And at this time, the estimates are pretty wide, but between all methane that including natural and anthropogenic, lakes produce about 2 to 10% of the global methane budget atmospherically. And this is really important. The processes that dictate whether or not methane is taken up by the food web or effluxed into the atmosphere are dependent on the lake characteristics. And a lot of small lakes, globally, the global distribution of lakes tends to be there's a lot of small lakes. In fact, 91% of the lakes are quite small. And these lakes tend to develop strong stratification. They also tend, in the boreal regions, to have high DOC concentrations. So there's a lot of lakes in the world that fit this criteria of a perfect place for methane to end up in the food web and be produced. A lot of what we know about methane dynamics in food webs comes from a little lake in Finland called Mecojärvi. And Mecojärvi has been the focal lake looking at a lot of work as how methane can be utilized in the pathways from methanogenesis into the food web. And it's pretty characteristic of a lot of lakes in Finland. 60% of the lakes in Finland are humic and are highly humic, meaning they have a high amount of DOC, dissolved organic carbon, which provides ample organic matter for methanogenesis to occur. They tend to be steeply stratified. They tend to be anoxic in the hypolimnion for long periods of time. So a lot of the lakes that we find, over 270 million worldwide, have the potential for some degree of methanotrophy, and some proportion of that, although I don't know what that proportion is, has the right conditions for methane to be a pretty important resource for both the microbial community and for grazers and the food web. These lakes that stratify steeply and have hypolimnetic anoxia tend to have low abundances of fish, especially when they are steeply stratified. So Mecojärvi has a huge hypolimnion by the time it's really well stratified. There's oxygen layers about 1 meter deep and 3 or 4 meters of hypolimnetic anoxia. And when that overturns in the fall, it leads to winter kills for fish. So the entire lake will go hypoxic or anoxic and lead to pretty strong bad conditions for fish. So these systems are naturally fishless a lot of times. And if there are fish, they tend to be growing pretty poorly. So one thing that we wanted to know is how the trophic structure of top-down control could affect methane dynamics in these systems. And so we wanted to know how fish could lead to how fish could modulate or exchange how rates of methane incorporation into the food web and how much methane is leaving the system to the atmosphere could change based on the food web dynamics. So we decided we focused on these perch here, and we found that if we really wanted to understand how the trophic dynamics could affect methane dynamics in the system, that adding fish might be a good approach. There's a strong precedence for this to have some merit. Trophic cascades are now a fairly central tenet to ecology, and we understand that the trophic structure of a system can very much dictate the biogeochemistry and the biogeochemical processes and fluxes of that system. So in a case where there's top-down control and an apex predator, if you will, maintains some regulatory factor on planktivorous fish, the zooplankton are relieved from that pressure and therefore can really knock down the amount of phytoplankton and primary production in the system. And that would be the clear side of this plot. Trophic cascades are very well understood and are pretty paramount in terms of how indirect effects of predation, indirect effects of top-down control can affect biogeochemical processes. When you don't have top-down control and you have an abundance of planktivorous fish, they can graze down the zooplankton to levels where they're ineffective at grazing phytoplankton, and you see a bloom. In this case, this is here. And there is a precedent as well for this to have an effect on efflux from lakes to the atmosphere, where the presence of fish and the presence of top-down control can actually dictate whether a system is autotrophic enough or has enough primary production to be a carbon dioxide sink rather than a carbon dioxide source. So there's a precedence for this. And so if this were true in a system that has methane dynamics like Mecojärvi or a lot of the systems that I've described today, what we'd expect is the presence of fish via a trophic cascade would lead to a change in the methane efflux from the system. When fish are present to graze zooplankton, which rely heavily on methane oxidizing bacteria, the methane oxidizing bacteria become abundant, they oxidize more methane, and we see less leaving the system. When fish are absent, zooplankton are proliferate and lead to reduced abundances of methane oxidizing bacteria, and these bacteria do not oxidize as much, leading to more methane. So we had a pretty testable hypothesis here, and we did what any other ecosystem ecologist with whole lake manipulation envy would do. We went out and we manipulated a whole lake. So to do that, we needed a long rope. Mecojärvi is a fairly small lake. It fits, I'd say, we could fit it into this row of chairs over here. It's about 0.3 hectares, so very small. So we needed about a 200-meter rope, and we needed to drag it across the whole thing and get a big curtain. And that big plastic curtain, we put floats on one side, and we weighted the bottom of it with sandbags. And then we drew straws to see who was going to step out on the ice first. And it's the only time I've ever been appreciative to not fit into an article of clothing, because that survival suit was really small. And my colleague Yari, it fit a little snug on him. So we sent him out there, and the first thing that happened is he put his leg through the ice. So we were a bit nervous, but in the end of the day, it worked out. Nobody went through the ice completely, maybe only up to our shoulders. But what we ended up with was a very well-placed curtain. And this curtain extended into the inflow and the outflow of the system so that we could have water come into the system equally across both basins. And we ended up with two really pretty equal treatment basins, and we applied fish to one side and not the other, and we measured zooplankton abundance, the abundance of methane oxidizing bacteria, the concentration of methane in the system throughout 3 years, and we alternated the basins. So in 1 year we added fish to one side, the next the other side, and so on. So we ended up with 3 years of treatment with essentially planktivory or no planktivory in terms of the fish presence. So lo and behold, we found out something pretty exciting, that fish eat zooplankton. This was I thought we could wrap it up and go home at the end of the day here, but turns out this is pretty well known. We saw pretty marked decreases in zooplankton abundance as soon as we added the fish. It was remarkable. So sure enough, as we added the fish, the zooplankton disappeared. It got to the point where at the end of the seasons, to sample the zooplankton for stable isotopes to see what they were eating, we had to sample thousands and thousands of liters to barely get enough sample to run a stable isotopic measurement on. So we saw pretty remarkable changes in the zooplankton abundance as we went. You can see here, this is what the samples looked like before we added fish. This is about a week after we added fish, so already a huge effect. And by the end of the season, there was no zooplankton to be found. These were our quantitative samples. The fish present basin was pretty easy to count. It took about no time at all. And to put it a little different way, when you look at the effect of fish after a small window after the addition, we saw really substantial declines, significantly reduced zooplankton abundance in the presence of fish. The change in zooplankton abundance led to a significant change in bacterial abundance as well. So we saw that immediately. We saw an effect. This is a pretty busy graph. It's showing all the bacterial abundance from the entire lake, but really what I want to focus on is the epilimnetic samples, where we see in the presence of fish, bacterial abundance went up, and when no fish were present, we saw lower bacterial abundances. So we saw pretty immediately an effect into the microbial community or into the microbial abundance data. When fish are present, we saw a change there. What we didn't see was a strong change in the bacterial community. So we didn't see any associated, any changes really with the community dynamic. We saw essentially the same community in the presence of fish or the absence of fish. The overall effect was a change in abundance. When we looked at methane oxidizing bacteria in particular, using quantitative PCR, we saw that there was a pretty dramatic change in the abundance of methanotrophic bacteria that correlated to the reduction in zooplankton. So when zooplankton are present and have time, they substantially reduce and significantly reduce the abundance of methanotrophic bacteria over time. Whereas if they're not present, we don't really see a big change in the methanotrophic abundance. So we didn't really see a huge increase in methanotrophic bacteria, but we didn't see a decrease when the fish were present, where we saw a huge decrease when no fish were present. So to really demonstrate that this trophic cascade has worked its way down through the food web into the microbial loop and added a strong biogeochemical effect, we had to look at methane efflux. And what we saw is that in the presence of fish, methane efflux to the atmosphere was about 10 times less than when fish were absent. So by adding fish, we therefore reduced the amount of methane leaving the system. One explanation might be that we just had spatially distinct methanogenesis. So maybe happened that when we added fish, the methanogenesis kicked up or something, or we saw a reduction in methanogenesis. But really what we found was equal methanogenesis. If you look at the hypolimnetic data on the lower panel here, we saw no differences in the methane concentration in the hypolimnetic waters, but we saw significant differences in the epilimnetic waters. So we saw the methanogenesis side of the balance between methanotrophy and methanogenesis didn't change due to our fish presence, but we saw major changes in the oxidation rates that as an artifact of the trophic manipulation we saw. So to really sum up the results, when there's no fish, there's lots of methane efflux in these systems. And when you add fish, you see a major change in about tenfold difference in the methane methane efflux from the system. That's a pretty substantial amount. So what we see here is that when you change the trophic structure of a system, just like in previous trophic cascade demonstrations, we saw marked changes in the biogeochemistry. And so what this really means is that the ecology matters. The ecology that surrounds what's going on, these biogeochemical processes, and the trophic structure that's setting the stage for these biogeochemical processes is really important. And the systems that have fish or don't have fish or have many species or many trophic levels of fish may have modulating and alternating effects in terms of the biogeochemistry. Further, we didn't see any really strong changes in the microbial community, just the abundance. So it's very likely that methane oxidation wasn't the only biogeochemical process affected by this. We saw just a reduction in the numbers of bacteria that were present, not any big swings in the biogeochemical potential. So it's likely things like phosphorus and nitrogen recycling from organic matter were affected by the presence of fish as well. And I really think that the understanding that the ecology matters is really what Lindeman's paper was saying, and I think this is another example of how he is well ahead of his time, and that he was absolutely right. The trophic dynamic is incredibly important, and it reaches deep into the biogeochemistry of at least in methane here, and likely lots of other potential changes. This has global significance. The fact that this lake in Finland had 10 times less methane when fish were present has a strong implication in terms of the global dynamic of methane when you consider that methane efflux can offset any carbon sinks in terrestrial systems. There's also some pretty important feedback mechanisms in terms of climate change that I think are pretty impressive. When you think about a warming climate and the fact that fish are increasing their range, moving northward, things like smallmouth bass could come into these systems that normally had very poor fish diversity or no fish at all, and lead to strong changes in the processes that I just described. So increasing ranges of fish northwards could have a pretty profound effect. Also, brownification is a major artifact of climate change in these systems, and what we're seeing is that as systems become more rich in DOC, higher organic matter in dissolved form in the lakes, there's higher potential for methanic genesis to occur, and that has big implications when you consider that methane is over 100 years 25 times more potent greenhouse gas than carbon dioxide. So the feedbacks here that we're talking about can really take hold and have a pretty profound effect on the overall processes here. Lastly, I think it's important to consider if a system has fish, those fish are doing something pretty important, and that the conservation efforts to maintain fish and native fish in particular this speaks to the fact that fish are important because their ecology is important, and fish conservation is a pretty important aspect when you consider what removal of fish or fish die-offs or introduced species could do to these dynamics, these biogeochemical dynamics and methane dynamics that I've described. And lastly, I think there's some hope in terms of the concepts that I described, that if we can have fish that can cause a tenfold decrease in the amount of methane emissions from lakes, there's maybe application in agriculture to consider adding fish to rice fields, where rice is a huge source of atmospheric methane. 10% of all the methane derived from agriculture comes from methane. So if we can reduce that 10% to 1% using trophic cascades, it would be a pretty important global process, and we'd see much reduced carbon or methane dynamics from using these concepts as a conservation or as an applied science. And with that, I think I'll leave you saying that in my 3 years in Finland, I learned one word of Finnish, and that's kiitos. It means thank you. I couldn't have explained it that well. There has to be questions.
What kind of fish would you add to rice paddies?
So the question was, what kind of fish would we add to rice paddies? So there's, I'm working with a group in California that has potentially looking into this as a climate change and a climate footprint mitigation strategy. And I think that the fish that they've talked about using so far is golden shiner, which are can really graze plankton down fast. So preferably native fish is what I would say. They've done some work looking at salmon, and they've done a fairly good job with Pacific salmon in the rice fields, but it wasn't scalable. All right, fire away.
You added planktivorous fish to your system. What would happen if you then added piscivorous fish?
I think that the title of the paper we talked about a lot, because essentially what we found is that if we add another layer, another trophic level on top, those effects would simply augment, that we would see the same concept take place, but instead of decreasing methane efflux, we'd see an increase in methane efflux. It might allow for more zooplankton, which would lead to less methanotrophic bacteria. So really it's that top level abundance that's driving whether or not a change in efflux occurs, or an increase or a decrease in efflux.
Thank you.
Right here?
Hi, yeah, the one in the slide that you showed with the two light, there was one lake with a partition with very different color. So I'm wondering if the change in general, does it have a strong optical signal?
Could you repeat that? I'm sorry.
Does it have a strong optical signal? When you divided your lake in half, did you have a strong optical signal between the two water masses?
No, we did not. We saw no change in the organic carbon dissolved form. We didn't see any major changes in nutrients, which I thought was a little odd. I was sort of hoping we'd see some kind of reverberation into the other biogeochemical processes that we could go after, but nothing really changed. I should mention that we did pull the curtain up between each season so that we re-equilibrated the system into two relatively equal basins, and at the start of each season we saw essentially equal everything on both sides. So we're really starting with the same template so I think that if there were potential for those kinds of changes to occur, we would have seen them. Nothing changed in terms of the color. The seki or transparency was still nearly non-existent. It was light is basically gone in this lake within a meter, so it was really, really dark. And we didn't see any change in that attribute.
Thank you very much for a very excellent talk. Your experimental system combines extremely high methane with a lot of anoxia and the unlikely presence of fish.
Right.
So I think the point is very well made. Now, what do you think, if you scale it to lakes that are not so extreme in terms of you need to have fish and methane for this to happen.
Right.
So how do you scale this to the rest of the world?
Yeah, I think that's an excellent point. We know that this is a bit of an extreme case with a lot of methanogenesis, a lot of potential oxidation to occur, and a system that normally doesn't have fish. So I think that our results I think you have to be careful simply extrapolating our results to every boral lake or every lake with a fair amount of organic matter, organic carbon in it. But it certainly, there's a gradient between an oligotrophic lake without these effects and something like Microbial. So I think that you just have to be careful when you extrapolate to other systems to know that these processes are likely happening in some fashion, whether it's methane oxidation or not. If zooplankton are relying on microbial carbon at all, it's likely that they're augmenting the biogeochemical processes that are occurring in that system. This was a good example of it. We were able to really pin it down and take good solid measurements to demonstrate that this is happening, but it's likely not exactly typical of most systems, I would say. All right, thank you very much.
Thank you, Shawn, for a wonderful talk, and congratulations again. Okay, thank you again for attending this plenary, and I just have a few things to mention before we adjourn. Tomorrow's award plenary is at the same time. We've got two presentations from the recipients of the Margalef Award for Excellence in Education and also the John Martin Award. And we have several events immediately following this plenary tonight that you should definitely consider attending. The first is the ASLO Fellows and Membership Reception just outside these doors, and there's an open bar. At 7:30 back in the ballroom is the ASLO Business Meeting. And then at 8:30 we have the Student Mixer, which is in the third floor foyer, and the Early Career Mixer, I believe that's on the fourth floor here. And one final announcement for any of those of you that may be here that attended yesterday's leadership workshop. John Downing would like you to meet him just outside the doors here to receive your ribbons now. All right, so thank you very much for attending. Enjoy your evening, and we look forward to seeing you tomorrow.